Hello friends, I'm going to discuss about cell membrane. What are the composition? They are carbohydrates, lipids and proteins. So carbohydrates occupy 3% of surface area and lipids 42% and proteins 55%. And lipids and proteins are in 1:1 ratio. First, carbohydrates. As I said, it occupy 3% of surface area. And there is a question, which constituent is responsible for asymmetry of the membrane? Options are carbohydrates or lipids or proteins or fatty acids. So the answer should be carbohydrates. Carbohydrates are arranged asymmetrically. And carbohydrates are responsible for immune reactions. Second, lipids. So which is the major constituent in lipids? That is phospholipids. And phospholipids, how will they behave at their low, moderate or high concentration? Let's see. Phospholipids at their low concentration, they form a single monolayer. Example, surfactant. And phospholipids at their moderate concentration, they form circular aggregates. It's like this. They form a missile like bile salt aggregates. (20 to 40 bile salt aggregates they form in a circular form, and the tail portion is fat soluble and the outward portion is water soluble and the fat substance is in between. So these are called micelles. Phospholipids at their high concentration, they form bilayer, that is our cell membrane. They form lipid bilayer. So this is a cell. This is our lipid bilayer. Hydrophobic tails are pointing inward and outside are the hydrophilic heads. And there is a question, when a cell is injured using a microneedle, what will happen? The options were lateral movement of the lipids or lateral movement of the proteins or enzymatic interactions or hydrophobic interactions. So when a cell is injured using a microneedle, hydrophobic interactions of the adjacent lipids happens. Hydrophobic interactions of the adjacent lipids, the process is called resealing. So our cell membrane is self-sealing through hydrophobic interactions. And then cholesterol. Cholesterol is the fluidity buffer of the cell membrane. So our cell membrane is called as fluid mosaic model or single Nicholson model. So cholesterol decides the cell membrane is fluid or in the solid state. Third, we are going to see about proteins. The first point here is proteins concentration varies from cell to cell. Protein concentration varies from cell to cell, and we will see how. So let's discuss. Let's see high lipid and low protein concentration. (76% of lipids and 24% of proteins So this ratio is seen in oligodendrocytes and in Schwann cells. Oligodendrocytes myelinates the CNS membrane and Schwann cell myelinates peripheral cells. And remember, myelin is lipid rich. Wherever the cell is myelinated, that is lipid rich and having high lipid content. I will give you an example. Nitrogen narcosis. When a person deep dive into the ocean, because of the high barometric pressure around the body, the gases will dissolve into the body tissues. So the main gas has been inhaled is nitrogen, right? So the nitrogen gets dissolved into the body, and nitrogen has high affinity for lipids. Since myelin is lipid rich and because of the high lipid content, there is a disturbance in the CNS membrane because the oligodendrocytes are myelinated, right? Because of the lipid rich content, this nitrogen gets dissolved in the cells and disturbance causing nitrogen narcosis we will see in the respiratory system. Next is high protein and less lipid concentration. And this is seen in inner mitochondrial membrane and presynaptic membrane. Inner mitochondrial membrane consists of 76% of protein and presynaptic membrane consists of 70% of protein. Inner mitochondrial membrane is having high protein because of the enzymes, and presynaptic membrane, you see this is the presynaptic membrane and here are the cells coated with myelin, right? And whenever there is a neuronal ending, it's devoid of myelin and having channels are with channels of protein based, right? So whenever there is a neuronal ending, it is devoid of myelin and less of lipid and more of protein. And this is how protein concentration varies from cell to cell. Let's see the second point, that is protein concentration varies from time to time. It depends on protein turnover rate of the cell. So this is the typical cell and here is the membrane protein. Membrane protein is synthesized inside a cell and it is coming to the cell membrane for its purpose. When its function has been done, it has to be removed and degraded, right? So it should be tagged first. The tagging is done by ubiquitins. Ubiquitins tags this membrane protein, then it's removed and it's going into the cell organelle. So which is this cell organelle where this membrane protein will be degraded? Options are ribosomes or lysosomes or proteasomes or Golgi bodies. So your answer should be 26, yes, proteasome. So a membrane protein is synthesized and it is going to the cell membrane, and when its function is done, it is tagged by ubiquitin and it is removed and degraded in the cell organelle, that is proteasome, right? This process is the protein turnover rate. If the degradation is faster than the rate of synthesis, the protein concentration will decline. So this is how the protein concentration varies from time to time. Example, sodium reabsorption. So this is I'm going to discuss the sodium reabsorption happening in the renal tubule, and sodium is coming, it's inside the lumen, and this is the apical membrane facing the lumen, and this is the basolateral membrane near the blood vessel. So when a sodium comes and it should be reabsorbed, right? The reabsorption, for reabsorption, aldosterone is the one helping. It is the protein channel called ENaC, epithelial sodium channel or amiloride sensitive channel, and this channel is protein rich, and here is the sodium potassium pump that is also protein rich. So whenever a sodium is coming into the lumen, it should be reabsorbed, right? It should be reabsorbed first. It's a two-step process. It should be reabsorbed here in the apical membrane, and it should be reabsorbed here in the basolateral membrane. The sodium here should be reabsorbed, then go into the blood vessel here. For reabsorption to happen, sodium reabsorption to happen, these two channels should be open, and it should not be removed or degraded. It should not be removed or degraded by ubiquitins. So what is the role of aldosterone here is de-ubiquinating, because if the ubiquitin tag this and remove this protein and degrade it, the sodium reabsorption will not happen. What aldosterone will do is it will de-ubiquinate and let the epithelial sodium channel to stay longer and increases the sodium reabsorption. Likewise, it increases the sodium potassium pump. So the sodium reabsorption increases as long as aldosterone is active. So the protein concentration varies from cell to cell, and protein concentration varies from time to time. This is all about the cell membrane, and let me know which topic I should discuss, and I